Hi, and welcome to the VieSID Press-release. In this video, we report on articles selected from the dental literature published in November and December 2023 that we hope might be of interest to the VieSID community. So let's begin. In Japanese Dental Science Review, a scoping review by Takahashi et al. Was published discussing the risk factors for cognitive impairment with focus on the decline in oral function. Cognitive impairment is considered an early stage of dementia. Dementia is the main reason for patients to require nursing care in Japan as in other regions of the world. According to the World Health Organization report, dementia is a syndrome characterized by a deterioration in memory, thinking, behavior, and ability to perform everyday activities. It has a physical, psychological, social, and economic impact, not only on people with the condition, but also on their carers, families, and society. The number of people with dementia was 50 million in 2015 and is projected to reach 78 million in 2030 and 139 million in 2050. There are many different forms of dementia, and Alzheimer's disease is the most common form, contributing up to 60-70% of the cases. Modifiable risk factors for cognitive decline at different stages of life have been identified. These consist in poor education during early life, hypertension, obesity, and hearing loss during midlife, and smoking, depression, physical inactivity, social isolation, and diabetes in late life. These factors might account for 35% of cases of cognitive impairment. In 2020, air pollution, excessive alcohol consumption, and traumatic brain injury were included to this list. Non-modifiable factors have been reported as risk factors for dementia. These include particular genetic variants of apolipoprotein in, amyloid precursor protein, and presenilin 1 and 2. Additionally, age and sex and estrogen reduction during menopause have been considered key factors. Protein markers, such as tau protein in the cerebrospinal fluid or peripheral blood related to neutrophil activity, have been found in individuals with dementia and Alzheimer's disease. Poor dental status is another recognized risk factor for cognitive impairment. Variables mostly considered are number of teeth and occlusal forces. A meta-analysis by Ho et al. In 2018 revealed that the high number of residual teeth was associated with a decreased risk of dementia of approximately 50%. Instead, poor oral hygiene, suboptimal oral health appear to be associated with increased risk of developing cognitive impairment and dementia. A meta-analysis by Ceruti Coplin et al. In 2017 showed that there are three possible mechanisms by which tooth loss can lead to cognitive decline: reduced sensory stimulation to the brain, poorer nutrition, neuroinflammation caused by periodontal diseases, and thus here tooth loss was thought to be the outcome of periodontal diseases rather than a risk factor in itself. In relation to stimulation of the brain, both animal and human studies showed an increase in cerebral blood flow and blood flow in the trigeminal nucleus in relation to mastication through exercises. Thus, chewing can be interpreted as a protective factor to cognitive decline. Animal studies showed the relationship between masticatory function and cognitive impairment. Reduced masticatory function led to reduced neurogenesis of the brain, according to Noble et al. Decreased occlusal support in mice might affect cognitive function by neural activity and neuroinflammation in the hippocampus and hypothalamus, based on results by Furukawa et al. Edentulous mice showed hippocampal neural cell loss, changes in hippocampal cell behavior and gene expression, as in Watanabe et al. In 2016, Takeshita et al. Reported that occlusion forces were significantly related to cognitive function. Occlusal forces, number of teeth, and periodontal status are mutually correlated, but periodontal status and the number of teeth were not significantly associated with cognitive function after adjusting for occlusal forces. These results suggested that occlusal forces may be the most important oral factor in relation to cognition. It is also known that people with lower occlusal forces tend to avoid vegetables and fruits and therefore habitually receive significantly less vitamins and fibers, which play an important role in protecting against cellular damage and chronic diseases, including cancer and cardiovascular diseases. Based on this review of the literature, the authors concluded that cognitive decline and dementia can be prevented by interventions aimed at reducing the known risk factors, including the oral factors. Thus, improved oral health and function through better oral hygiene and oral rehabilitation should be the target of healthcare policies as one of the ways to prevent cognitive diseases. In the same journal, a further paper on a similar subject was published by Stamaria et al., investigating the relationships between mastication and cognitive function by means of a systematic review and meta-analysis. The authors based their study on the assumption that tooth loss can lead to changes negatively affecting cognition, including changes in diet and lifestyle and diminished chewing performance, leading to decreased cerebral blood volume and reduced brain activity. Instead, mastication activates brain function and directly stimulates the hippocampus, thereby increasing the activity of the neurons responsible for memory and cognitive function. Cerebral blood volume increases, providing a greater oxygen supply beneficial for promoting brain cell activity. For their review, they proceeded by screening the literature published between 2000 and 2020 and eventually considered 20 articles that studied the relationship between cognition and mastication by established protocols in the elderly, using, for example, the mini mental state examination and other neuropsychological tests on attention, working memory, and verbal fluency. And then consider also additional studies focusing on younger adults, where cognitive function was assessed considering the processing speed of cognitive tasks. Therefore, the reaction time was considered in relation to chewing. In particular, the authors aim to address the following two main research questions. The first, is the cognitive status of older adults with dementia associated with masticatory function? The second, does cognitive function improve with mastication in young adults? Here I report a short summary of the results by Stamaria et al. The authors interpreted their results cautiously, as the evidence they included in their work was also of poor and fair quality. However, they concluded that cognitive function tests were significantly lower in subjects with lower masticatory performance and chewing ability and a decreased number of teeth. The risk of cognitive impairment was higher in subjects with masticatory dysfunction, such as chewing difficulties, decreased masticatory performance, low chewing ability, and a reduced number of teeth. Mastication reduced the cognitive processing speed, meaning improved the reaction time of young adults when performing cognitive tasks. The Journal of Dental Research published a scoping review by Sangalli et al. discussing the importance of sex dimorphism in oral health and reporting on sexual variation in the prevalence of the main oral diseases. In the last few decades, the importance of sexual dimorphism has been appreciated in the medical field, which reflected in an increasing number of publications taking into account sex as a biological variable. Biological sex can be defined categorically as a distinction between male or female, genetically as characterization through a distinct set of sexual chromosomes, XY or XX. Let me also add here that this classification does not take into account sexual chromosome abnormalities, such as, for example, Turner syndrome, or XYY male, or XY female. There is also phenotypic classification based on classification into males and females, relying on morphological characteristics. Sex, however, should not be confused with gender. Gender can be defined as a social construct that refers to an individual's identity, self-perception, and self-identification in sex-related roles in response to cultural influences, social roles, and expectations. Although it is not easy to completely disentangle the effects of physiological, psychological, and social factors in the development and expression of oral pathologies, the authors reported on the current knowledge of the pathophysiology underlying differences in oral health between males and females. Gonadal hormones are known to play an important role in the predisposition to periodontal diseases. Low-dose estrogen induces pro-inflammatory cytokines, bone loss, and gingival inflammation, while high-dose estrogens, as during pregnancy or menstrual cycle, have been related to decreased inflammatory cytokine production, although periodontal diseases might worsen during pregnancy. Androgens were significantly associated with higher cancer prevalence, while estrogens seems to have a protective effect. Gonadal hormones are also implicated in pain perception. Genetic factors. The female predisposition to caries was suggested in relation to specific genes located on the X chromosome involved in amelogenesis, salivary flow, diet preferences, and oral microbiome. Of genetic origin are also the differences in immune response and perception of pain. Immune response. Men display a stronger innate inflammatory response to injury and microbial pathogens than women, which might explain the higher prevalence of periodontal diseases in men. Females show more robust innate and adaptive immune system, making them prone to autoimmune diseases. Gender has been shown to influence behaviors linked to manifestation of pain, as society norms seem to allow female to express pain, while male expression of pain is less tolerated. For example, individuals of masculine gender showed higher tolerance to pain if the investigator was a female. Psychological conditions such as anxiety, depression, stress response, and coping versus catastrophizing are known to influence the perception of pain and reaction to diseases and dysfunctions. Social factors, including marital and socioeconomic status and education, as well as drinking and smoking and dietary habits, are known to greatly influence oral health. Differences in the physiological factors just explained translate in differences in the prevalence of oral diseases. Periodontal diseases are more prevalent in males than females, despite the higher female propensity for tooth loss and edentulism. Systemic factors such as diabetes and obesity, more prevalent in males, play an important role in the development of periodontal diseases. Conversely, females are more prone to periodontal diseases during puberty, pregnancy, and postmenopause, which trigger an exaggerated inflammatory response of the periodontal tissues. These observations apply to both gingivitis and periodontitis, although evidence for grade C periodontitis is contradictory. Caries are more prevalent in females than in males, and discrepancies between sexes increase with age. Possible factors that negatively influence caries in females are earlier tooth eruption, pregnancy, and possibly access to snacks while cooking, depending on the habits. The discrepancy in occurrence of caries exists despite males generally brush their teeth not as often as females. Apical periodontitis seems to be positively associated to endodontic treatment, age, and presence of systemic conditions. However, evidence of sex-related differences in the prevalence of apical periodontitis is inconclusive. Nonetheless, there seems to be a sex-related difference in the response to apical periodontitis. For example, apical periodontitis has been shown to be significantly more prevalent in diabetic men than in diabetic women. Women with inflammatory bowel disease show higher prevalence than control, but this was not observed in males. And women who smoke are more affected than men who smoke. Most of the orofacial pain disorders are more prevalent in females than males. Many orofacial pain conditions are comorbid with systemic and autoimmune diseases, known to be more common among females. Higher prevalence in females manifests after puberty, while these differences between males and females disappear after menopause. Thus, an influence of gonadal hormones is likely. Oral cancer is much more prevalent in males than females, even to a ratio of 5:1. Higher prevalence in males remain even when accounting for modifiable risk factors such as smoking and alcohol consumption. In conclusion, I suppose that for each of the discussed oral diseases, the influence of biological sex is certainly more complex and profound than it could be presented in this paper. However, this article by Sangali and colleagues brought an important topic to our attention, that of sex and gender-related variation as relevant factors to be considered when discussing the pathogenesis of oral diseases. Certainly a topic deserving attention both in scientific and clinical settings. Accuracy in the presence of implants. First, the authors identify the known parameters affecting intraoral accuracy, which can be classified as operator-dependent or patient-dependent. The operator factors include device technology and system, scanning head size, calibration, ambient lighting in luminance conditions, scanning pattern, experience of the operator, cutting off, rescanning and overlapping techniques, extension of the scan, and scanning depth and angulation. Patient-related factors include the intraoral conditions of the patient being scanned, such as tooth type, interdental spaces, arch width, palate characteristics, wetness, existing restorations, their geometry and characteristics of tooth preparation, and presence of edentulous areas. The authors discuss some of the parameters more in detail. For example, they recommended following the instruction of the manufacturer in terms of ideal lighting conditions and using a luxometer to verify that scanning is operated under the correct lighting. Possibly the scan should be performed in an environment without windows to better control luminosity, as deviations in lighting conditions might lead to decrease in accuracy ranging from 20 to 250 micrometers. They recommend the scanning pattern varying between different devices should be followed. However, the authors also noted that these patterns are tested on dentated patients and not on edentulous patients or implants. It is known that accuracy varies depending on implant body design, including the material, geometry, and retention system as possible factors. It seems that metallic implant scan bodies results in higher scan quality than polymer implants. Scan body splinting techniques have been suggested to improve the scan quality, but as noted by Revilla Leone and colleagues, evidence is too scant for conclusive statements. With regard to the patient's related parameters, I will make a very short summary of the accurate report of Revilla Leone et al. Arch location. It is not yet clear whether accuracy differs between maxillary or mandibular model, and to what extent. The studies were performed in vitro, and the palate was missing in the maxillary models. Implant position. Although a quantification is difficult, it is known that the more distal the implant, the lower is the accuracy of its representation for the known distortions occurring in the posterior regions of the intraoral scans. Also, if bilateral implants are present, the one that is at the opposite side of the side where the scan starts presents higher distortion. Interimplant distance. The longer the distance between implants, or in other words, the longer the non-dentated areas, the lower is the accuracy of the scan. Angulation in implant depth had also been considered, but the outcomes are contradictory. Interdental space or space between tooth and implant can affect the quality of the scan when there is not enough space to actually represent the entire implant, as in the case represented here. In conclusion, how to improve the accuracy of the intraoral scanning in general? Recommendation number one is to follow the manufacturer's suggested parameters. Of course, operator experience plays an important role here. These recommendations, of course, apply in case implants are present. However, the presence of implants, or let's say the lack of teeth, adds to the risk of inaccuracy and distortions. There is no convincing evidence on how to improve the quality of intraoral digital implant scans, although it seems that metallic implant scan bodies are preferred over polymer implants, and implant scan body splitting methods may improve the accuracy of intraoral digital implant scans. We've come to the end of this presentation. I invite you to contact us in case you want to know more about our activities. Otherwise, be ready for next press release. Best wishes to all.